I hope the screen is visible. So, hello, good afternoon. Welcome to week 3 of the discussion session for the immunology course. Now, as you are already familiar, in these discussion sessions, we go over some of the topics that have already been discussed in the past week. And we try to address some of the questions that are related to to the topics that have been covered for that week. Now, as you are already aware, for the previous week assignment, since the deadline was on 7th, we had not discussed those assignments beforehand. So that is what we'll start off with. We'll discuss the questions that were given there. I'll go over the correct answers. And in case anybody had any queries regarding it, we can go over it again. We can try to solve those if anybody has any doubts, or we can just have a discussion about it in general. Apart from that, since this week's topics were majorly centered around the structure of the antibody and the way in which antibodies are synthesized, not in terms of the cells that undergo the process, but rather the molecular mechanism that operates in the generation of antibodies or B cell receptors, as they are more correctly termed. So we'll cover those topics very briefly only because it has been extensively discussed in those lectures. So there are some additional information that were not a part of those lectures, but you will come across if you go through the reference book suggested by the professors. So we'll cover some of those experiments as well, which led to the discovery which we are studying as part of this course. Um, Apart from that, at any point of time, if there's any doubt, please let me know. You can interrupt me at any point of time, and we'll take up questions. At least we'll make a note of what needs to be discussed in the next sessions. So with this in mind, I'll start the discussion for today. So first, we'll go over the questions that were given as part of week 1 assignment. Just a So, as part of the questions, the first question here is about the origin of hematopoietic stem cells. Where do they originate? So the options are bone marrow, spleen, liver, and fetal tissues. Now, this is a question that can be very easy to get wrong because you think of hematopoietic stem cells and you immediately answer bone marrow. And in fact, that is the way we have discussed the development of B cells and T cells as well in the discussion sessions and also in the uploaded lectures. But what you need to keep in mind is this is about the origin of HSCs, not about the origin of the immune cells from the HSCs. So their development, in fact, the origin of stem cells begins at the time right after fertilization has been completed and developmental processes have started to operate. That is when stem cells are synthesized. All kinds of stem cells are synthesized from the embryonic stem cells, which are ESCs. All these different subtypes of multipotent or multipotent cells or stem cells are synthesized. So hematopoietic stem cells are an example of multipotent stem cells, and their origin will, of course, be credited to fetal tissues. Once it has been synthesized, the hematopoietic stem cells will lie within the bone marrow, and there they will give rise to all the different lineages that we have extensively been discussing since the first week. So again, it's just about a very it's about being a little careful when you answer questions like this. You make sure you read the question again so that you don't get it wrong. If there's any doubt, again, please interrupt me. You can raise your hand or interrupt me at any point of time. Unmute and ask your question. Question 2 is about which of the following cells form the innate immune system. So we had discussed the different types of cells and how some of them are overlapping between the innate and adaptive branch, how there is humoral immunity, cell-mediated immunity, how there's a coordination between the two branches, information from innate is being directed to adaptive. So all of this has been covered. So let us look at the options. The first option is about B and T cells. B is macrophages and NK cells or natural killer cells, both A and B or none of the above. Now, if you have attended the previous lectures or the discussion sessions, you would remember that B and T cells are, of course, the lymphocytes that take over the majorly take over or majorly mediate the adaptive branch of the innate adaptive branch of immunity, which comes in as a later response that kicks in to tackle a pathogen that cannot be handled alone by the innate immune system. So they mediate majorly the adaptive branch. On the other hand, innate immune system is taken care of by the macrophages and NK cells. Mainly, there are also other subtypes that come in, but from the options given, option B would be correct. Uh, Third question is about the types of cells that are present in the spleen. So if you remember, we had looked at the cross section of all of these lymphoid organs, the primary and the secondary lymphoid organs and the lymph nodes, etc. We had gone over these. You can refer to these slides in case there is any confusion. And of course, you can refer to any textbook or even for that matter, you can go through research papers if you have that advanced habit of reading research papers. You can go through those as well. So if you remember, we had gone through the spleen and its different regions, how the localization of different cell types mediates the development of B cells and rather the maturation of B cells. So it is not solely by B cells within the germinal centers, but there are also other different cell types. So keeping all of that in mind, the options here talk about T cells, B cells, macrophages, or all of the above. Now within the spleen, or with the particularly the marginal zone of the spleen, all of the options are correct, and therefore B cells, T cells, and macrophages are present. So option D was the correct answer. Uh, for question 4, lymphoid progenitor cells do not give rise to which of these following cell types? So if you remember, when we again discussed the development of different cell subtypes from the hematopoietic stem cells, we had talked about the myeloid progenitor and the lymphoid progenitor. The lymphoid progenitor very it obviously gives rise to the lymphocytes and also to dendritic cells. If you can again refer to the older slides, that dendritic cells could be formed as a result of maturation from either the lymphoid or the myeloid progenitor in both cases, and therefore they were said to be overlapping between the two branches as well. But cell types that are not covered or that cannot be derived from the lymphoid progenitors are the granulocytes. So dendritic cells obviously can be generated by both. B lymphocytes are definitely derived from the lymphoid progenitor. Granulocytes cannot be generated from the same, and therefore the correct option was granulocytes. Um, in question 5, we were asked about circulating lymphocytes and where they encounter their antigens. So if you remember, we had talked about the fact that the origin of lymphocytes, their maturation, and the place where a naive cell will encounter its antigen and then become clonally selected and then clonally expanded. All of these occur in different locations. So when we talk about circulating lymphocytes, the region where they interact with their cognate antigens or where they face their cognate antigens is not in the primary lymphoid organs. It can be, but very rarely that happens because the pathogen is usually it will not directly enter the thymus or the bone marrow or the spleen for that matter. Instead, it will usually encounter it somewhere else when it is circulating within the body, and those are usually the peripheral lymphoid organs, or even particularly if we can be a little more specific within the lymph nodes. So option B in this case is the correct option. Uh, Question 6 is about Peyer's patches of the intestine. They are a part of Galt, Bolt, or Nolt. So among them, it is a part of Galt or gut-associated lymphoid tissue. In the first week's discussion session, there was a schematic representation of all of the different types of secondary lymphoid organs, where they're located, what they're called. So you can look into that. And also, of course, you can look at reference books for more details about it. I don't think I discussed Galt in much detail, but yeah, they're part. Peyer's patches of the intestine are considered to be a part of the gut-associated lymphoid tissue. Um, then we had a question where we had to match some different subtypes. So when you encounter such questions, obviously because you see that the number of options on either side, while they look equal, you can see that there is some commonality between some options. For example, if we look at antigen presentation, yeah. If we look at antigen presentation, for example, we know that all types of antigen presenting cells are capable of doing that. So that would be macrophage, dendritic cells, and if B cells were there, then them as well. So it's a multi combination options, and you can get questions like this. Definitely in the examination for this course, but also if you are planning to appear for some sort of an all India examination for PhD or for masters, etc., you will find questions like these. So in that case, A, of course, read the question carefully. B, you should opt for the process of elimination. You go for the, like for example, if we look here, of course you need to have some basic knowledge, but also go for the process of elimination. So for example, if you just look at the options overall, give it a glance, you will see that in some cases you will find some common options which are present in all of the, some common matchings which are present in all of the options. And with that, you take in your basic knowledge and see how you can eliminate this process. Now, because it would be difficult to show why the other options are not correct, I'll go through option B directly. So macrophages are obviously, they have been linked to 2 and 3, which means they are capable of antigen presentation. We know that. And also for phagocytosis, they engulf cells, and they are also responsible for killing bacteria, or bactericidal mechanisms can be activated. For option 2, we have dendritic cells, obviously again antigen presenters, but they are also responsible for taking up antigens in peripheral sites. Dendritic cells do not mediate killing of antibody-coated parasites. No, they do not. They are not granulocytes. They cannot release granules, and they do not promote allergic responses. So obviously, those are not correct. If you just go over the options, obviously, this release of granules, or what we had talked about as an inflammatory response very briefly in one of the initial lectures. This release of granules can be either, it is usually by mast cells, or it can in some cases be by macrophages, by mast cells. In some cases, some other cells can also come in, but it needs to be a granulocyte in nature. So this would be the correct option in that case. So from there you get BCD. So in this way you can eliminate any incorrect options. And the final matchings would be for neutrophils. They are responsible for phagocytosis only among all the other options. For basophils, we have allergic response promotion, and anti-parasitic immunity is also somewhat mediated by them. We have eosinophils, which are responsible for killing of antibody-coated parasites. So eosinophil action is majorly considered to be an antiparasitic response only, whereas basophils are somewhat mediated in the hyperinflammatory response. Not hyper, just an inflammatory response, and also for antiparasitic immunity to some extent. So in this way, option B was correct in this case. Next question was about B cells and how they mature into antibody producing cells in which part. So within the B cell corona, marginal zones, and germinal centers, red bulb. So you might not be familiar with all of the options. It's not even necessary. What you just need to have the conceptual soundness in this case. So we talked about germinal centers being present towards the cortical region, and how they are the sites of hyperactive B cell maturation, where it was occurring very quickly and very randomly, and we went through the entire process of how centroblast formation occurs, and how the selection with the antigen occurs, and how TH cells have a role to play, and the B cell that has the highest avidity or highest affinity for its antigen, only they get selected. So you get an optimized circulation of antibodies. In case you cannot recall what I'm talking about, I'll just show you the image. This was the entire process that we had talked about, how these cells develop and what roles they have to play. We extensively discussed the events that occur within the germinal centers. So you can go over those slides once again, just to brush up the topics if there is difficult to understand. So this maturation is obviously complete within the germinal centers. Um, then the next question was about Treg cells. Now, I don't think I discussed Treg cells directly, but if you remember, it was part of the lectures, and there you got information about how Treg cells, what are the different subtypes of T cells, first of all, and what role T regulatory or Treg cells have to play. So let's go over the options. First option was about killing other cells infected with viruses or intracellular pathogens bearing the antigen. Second, to provide signals, often in the form of specific cytokines that mediate the functions of other cells, or to suppress the activity of other lymphocytes and help to limit the possible damage of immune responses. Now, when we talk about the first two options, those are actually taken care of by a number of other cells, TC cells and T helper cells, which are the other subtypes of T cells, and they are extremely crucial in mediating the immune response as well. But Tregs are a more subdued subtype of immune cells or T lymphocytes, and their job is to regulate, control, or in fact, as the option suggests, to suppress the activity of other lymphocytes and therefore control the extent of damage that a non-specific or incorrect immune response can generate. So option C is the correct option in this case. The other two are for TC and TH, and of course, all of the above is not the correct option in this case. And finally, week one assignment question was about identifying the correct event sequence. So antigen presentation. So this was the, if you remember, there was a schematic I had shown for this as well, the sequential flow chart of events that occurred for antigen presentation and the way in which its recognition is occurring. So antigen presentation is, I'm directly going to the correct option, is carried out by APCs or antigen presenting cells that gets recognized by lymphocytes. That lymphocyte will then differentiate into a lymphoblast, which can occur, which can undergo very quick, very rapid proliferation into daughter cells, each of which will carry specificity with the for the same antigen in the same manner. And then from those daughter cells, they will undergo differentiation into effector cells, which can release the or secrete the antibodies that can go into circulation, and some of them will stay behind as the memory cells. So that was the entire process of events that occurred. If you look at the rest of the options, there is some switching that has occurred in this sequence of events, and therefore the correct option was option one. So I hope this was clear. If in case there is any doubt, please don't hesitate. You can go ahead and ask me, and I'll try to address your question to the best of my abilities. Uh, for week two, we had a different set of questions. The defensive barriers of innate immunity are, we went into more specifics about innate immunity, but yeah, the defensive barriers of innate immunity is or are anatomic, physiologic, phagocytic, and inflammatory. If you remember, again, there was a table that showed this, and of these, all of the options are actually correct, and they are all different levels of regulation of the innate immune system, and therefore the correct option was option E. Second question is about the receptors which help dendritic cells bind microbes and carry them to their secondary lymphoid tissues. So if you remember, we had talked about how innate immune system is being regulated. What, apart from these cell types, what are the different receptor types or the recognition moieties which are able to identify something as foreign, and in response to that identification, they are able to trigger an immune response, or they're able to trigger um, an event which will alert the system that something foreign has come in. So that set of receptors were called as pattern recognition receptors. And we looked at different subtypes under that as well, for example, Toll-like receptors, etc. So PRRs, or pattern recognition receptors, are the correct answer to this question. Then come the cell adhesion molecules, or CAMs, that are expressed on the surface of endothelial cells, and what are they known as. So if you remember, we had talked about in quite extensive detail that in case there is some injury within some tissue, then the way in which neutrophils will rush to that site of injury and they will regulate a response to it. So there were several sub-steps, how there is rolling, adhesion, what are the different interactions that occur between selectins, IgAMs, etc. All of that is there in the slides. You can brush up on those topics once again. But to address the question, when we talk about endothelial cells, the types of cell adhesion molecules that are present on them are selectins, L-selectin in particular. The other options are mucins, integrins, and ICAMs. They are also present, but with respect to this question, selectins is the correct option. Then come the which of the following are the cell types involved in innate immunity? So we had done a similar question in the previous week also. So there the correct options also included natural killer cells along with macrophages. So the cell types involved in innate immunity in this case would be natural killer cells, because B cells and T cells are obviously a part of adaptive immunity. So option A is the correct answer. Then in the next question, we were asked which of these are the first cells to be recruited at the site of infection. I just said it like just a minute ago that we had discussed the way in which at the site of injury, the first cells to rush to that site are neutrophils. They undergo that process of extravasating from the tissue, going into circulation, etc. All of that has been discussed in a lot of detail. So neutrophils or option B was the correct option in this case. Uh, T cell maturation occurs in the bone marrow, spleen, thymus, or liver. So, if you remember again that we had discussed the way in which B cell and T cell maturation also occurs. T cell maturation was discussed in less detail, but yeah, it occurs within the thymus. That would be the correct option. Rapidly dividing B cells in the dark zone of germinal centers are called, if you remember, I just showed that schematic. And then we saw that within the germinal center dark zone, those cells that are undergoing very rapid division are the centroblasts. Then they face the antigen that is displayed by dendritic cells. And following that, they would undergo selection for those with high affinity and they would be released into circulation. So centroblasts are the correct option. The other options are not true. They are centrocytes, plasma B cells, or plasmablasts. They are not correct. It is centroblasts. Uh, Next question was about how B cells interact with dash cells in the light zone of the germinal center. So if you remember, obviously they interact with dendritic cells, but also T helper cells. Again, referring back to that schematic. So here you see the follicular dendritic cell that is present at the center and how TH cell is helping with the stimulation as well to allow for the selected centrocyte from the centroblast. And then it differentiates into a plasmablast or a memory T cell. So among the sequence of events, it is interacting with these two. So keeping this in mind, yeah, the correct option would obviously be both A and B or option C. CD40L is present on the surface of B cells, cytotoxic T cells, or macrophages or helper T cells. So if you remember, there is a schematic for this also in most probably the discussion session for first week, where we had compared the type of cell surface receptor present in THTC and B cells. And therefore, CD40 was present, CD40 ligand was present on the surface of T helper cells or T cells. This is a fact-based question, so you can address it directly. And finally, for I guess yeah, the last question for week 2 was about granzymes. So they are secreted by cytotoxic T cells, T helper cells, macrophages, or B cells. So granzymes are by cytotoxic T cells, and they are helpful in killing of the pathogen that has come in. So that is the correct option. So with this in mind, these were the questions that were given. I hope you were able to address them correctly. If not, don't worry about it. As you attempt more and more questions, you will gain confidence about on how to address questions like this and how to do it as quickly as possible without spending too much time in case your goal involves some sort of a competitive exam in the future. So yeah, with this in mind, we move ahead. Again, if there's any query, please you address it here. Or if you want, you can also put it up on the discussion forum. And there somebody, some other coordinator for the course can help you address that question. Now, with this in mind, we will go over a brief recap along with some additional information to help understand the already discussed concepts better. If there is any doubt, please ask me a question. You can raise your hand or interrupt at any point of time. There is no, don't hesitate. Okay. So, so the first and second weeks, apart from an overview of the way in which the immune system works, the second week basically introduced the concept of B cell and T cell receptors. And as we have extensively discussed, the B cell receptors are the antibodies that we all are all too familiar with. And what we are looking into today are more details about the antibody that we know of. So on the left, you see a schematic of the easiest representation of an antibody. I'm saying easiest, it does not look very simple, but yeah, it's the easiest representation that shows how it actually looks in 3D. So what you see again, obviously, is the Y-shaped structure. You will find both types of representations. In some cases, the light chain is drawn, demonstrated on the inside. In some cases, it is demonstrated on the outside. It's shown in both ways. In 3 dimensions, obviously, if you remember Sir's lecture, he had shown that they crisscross. They are not present as two separate entities, but the two peptide molecules, they crisscross so as to form a good pocket where it can bind very well with its cognate antigen. However, of course, the antigen binding site is present right here at the top, which is the epitope of an antibody. So I don't have a slide for it, but just to clear some terminologies, any antigen antibody interaction has two participating moieties. A moiety basically means entity or any participant. Let's call it a participant for now on. Any surface or any entity is a moiety. So the two participating entities are epitope and paratope. So the part of the antibody that mediates that antigen antibody interaction is the epitope. So we say epitope of an antibody, and what it interacts with, for example, some protein, if it binds to here, the part that it interacts with within that protein will be its paratope. So epitope paratope interaction mediates the antigen antibody interaction. So with this in mind, yeah, somebody has a question. Please go ahead. Gauri, please ask your question. You can type it out if there's any doubt, because I cannot hear you. Okay, if not, you can interrupt me sometime ahead also, no worries. Uh, Coming back here. So we were talking about the antibody structure. So what you see here is a representation where the light chains are shown on the inside. What else do you see? You see two longer molecules or longer lines, which are the heavy chains, and you see two shorter lines, which are obviously the light chains. The light and heavy name comes from the fact that if you separate these molecules, their molecular weight or their length, based on that, you can say that these are heavier or lighter molecules. Among them, broadly speaking, you already this has been discussed in detail, so I'm going over it a little quickly. What you see is that it can be broadly classified into two subparts. The antigen binding, or FAB, which indicates fragment antigen binding, and FC, which is fragment crystallizable. It's the part that has been crystallized. And this part, FAB or FAB, otherwise known as FAB, is the part which is necessary and crucial for antigen binding. But antigen binding is not the only job of an antibody, right? It has just recognized an antigen. In response to the recognition, the signaling pathways that downstream need to be stimulated are taken care of by the lower region, which is why it's said to mediate biological activity. What you see here, these little loops, this is very commonly called as the immunoglobulin fold. Why so? Because it is an if you have studied protein structure previously, you would know about alpha helices, beta sheets, and how these secondary structures they help in forming three-dimensional structures, motors, domains, etc. If not, you can go over these topics also for your own preference. One such fold, very commonly known fold, is the immunoglobulin fold, otherwise known as the IG fold. So this is representative of an IG fold. It has this link where you can see that around 100 to 110, 120 amino acids approximately lie within this fold, and they mediate the structure, well-identified structure of an antibody. What else do we notice here? We see that there are some linkages which are present. What are those linkages? Most abundant linkage is the disulfide linkage. If you're not already familiar with it, it is most commonly observed between amino acids that contain a sulfur residue. When an amino acid is present by itself in isolation under physiological conditions, its side chain has SH or sulfhydryl group, and when two such amino acids come into close contact with each other, and there is removal of water from that region, those two sulfur molecules can form a linkage, a very strong linkage, which is very abundant in most proteins. And it's a very relevant linkage also, because it adds to the stability of a protein. That is known as the disulfide linkage. And the participating amino acids are most commonly cysteine. So two cysteine molecules form cysteine, or a disulfide bridge, or a disulfide linkage. So that's what you see here, that within each amino acid fold as well, you see these, within each immunoglobulin fold as well, you see these disulfide linkages, and you see some disulfide linkages between light chain and heavy chain, and also between the two heavy chains as well. So all of that holds the antibody structure together. So That's what we see majorly. And there are interchain disulfide bonds, as is shown, as we just discussed, and some intra-chain disulfide bonds as well. Apart from that, the nomenclature is quite obvious. You have VL and CL, which is the variable region of light chain, and the constant region of light chain. You have VH, which is the variable region of heavy chain, and CH1, CH2, CH3, otherwise also known as CDR1, CDR2, CDR3. These are the CH1, CH2, CH3 are the constant regions, and within the variable region of the heavy chain and light chain, which form the antibodies main recognition site for the antigen. Otherwise, what we talked about as the just now talked about as the epitope of an antibody, they contain these regions, these small regions that you can see highlighted here in yellow, which are called the hypervariable regions. And those hypervariable regions are called as CDRs, or complementarity determining regions. I just confused it a little bit. If there's confusion, please let me know. I'll clear it up again. So CDR1, CDR2, CDR3, etc. are the regions of hypervariation, are very if you remember that graph, it showed that these are some regions which have very less consensus, and any random amino acid can be located within them. Apart from that, you see some cleavage site as well. This is where papain can cleave. This is the hinge region of the antibody. Pepsin can cleave here. Complement binds to these regions. And here some additional carbohydrate molecules are also present, which helps in increasing the solubility of antibodies. Why do we need solubility of antibodies? That's because when they are in circulation, it helps in keep their it helps them to stay stable in the circulation. So that is overall the structure of the antibody. Next, what you see on the right is a diagrammatic representation of the two concepts that I personally had a lot of difficulty with when I was initially studying immunology during my college days. And there, I had a lot of confusion understanding the difference between affinity and avidity. But as has been explained very well in the lecture as well, and also I found a good schematic to clarify that concept. Affinity obviously is easy to understand. Affinity is something you can just mathematically calculate. It refers to the strength of a single antibody antigen interaction. For example, here you see each IgG antigen binding site typically has very high affinity for its target. It means that if there's a certain concentration of antibodies in a individual's system, for example, in my system, then of those antibodies, how many of them are able to bind to their antigen and how many of them are free, and then you take a concentration of those two. So that's easy to understand. If more and more antibodies are bound to the antigen, that means they are very strong, their interaction is very strong, and therefore this antibody has very high affinity for its particular antigen. And when it has high affinity, it is something that will be chosen by the immune system under most circumstances to be expanded so that more and more specific recognition can be carried out and the antigen can be removed from system, or the pathogen at large can be cleared off from the system and we become disease free. I still don't know what Gauri has to say. Your hand is still raised. If there's a question, you please go ahead and ask. Uh, on the other hand, what you see on the right is a representation of avidity. Now, as Sir had explained in the lecture, avidity represents the sum total of all of the interactions that are occurring. So, for example, when you talk about an antibody and its two arms, then you take the sum total of those two interactions. And if in case the sum total of those two interactions is stronger than another antibody's interaction alone, then it would have higher avidity. Yeah, Gauri, please go ahead.
Can you hear me?
Yeah, I can hear you now.
Ma'am, from where can we access these slides?
So you would have access to a Google Sheet most probably. So our YouTube videos are uploaded there, and there's a Google Drive link also. So that Google Drive link contains all these additional information. You don't have access to it. I found a lot of people trying to access the Google Sheet when I had initially forgotten to share the link. So I think people can access it. You can check with the course instructors, not instructors, but the coordinators. You would have access to a Google Sheet from all of the TAs, not just me. I think there are 3 people who are TAs on this course. So everybody uploads their slides. So you can get it from there.
Okay, ma'am, thank you.
If not, you let me know. I'll maybe include it on my on the YouTube channel where I upload the videos. But it's usually not advisable because this is part of NIPTIL and that is on a public platform. But yeah, maybe I can upload it there so you can access it from there. So yeah, coming back to the concept of avidity. So avidity was the sum total of strength of all of the interactions that were combined. So here you see a representation of a different subtype or a different isotype of antibody, which is IgM. So IgM has it's pentavalent in nature, which means that it forms a structure where 5 antibodies all together are able to bind to a particular antigen. This is the way in which it is structured. Its structure is maintained. So as a result, while IgM usually has low affinity, that each epitope paratope interaction might have less strength, but because there are 5 antibodies and as a result it has 10 binding sites, the net avidity of an IgM molecule is much higher. So you will get more details on this when we start understanding why isotype switch happens, how it is chosen, which type of antibody should come in, and so on and so forth. So we'll get into that later. But this just explains the concept in which avidity and affinity and avidity, both of them work. So with this in mind, now we come to the development of antibodies. So if we go through the overall process, first of all, you have the hematopoietic stem cells naturally, where IgG is not expressed. Here on the right, you see a comparison whether or not an immunoglobulin is being expressed at any stage. From the hematopoietic stem cell, you see this blue arrow, which indicates that it is capable of replicating itself. So stem cells have the capacity of self-replenishment, which means they are able to divide and self-dividing without any additional stimulus. From there, they can differentiate into the lymphoid cell progenitor. They will undergo some partial heavy chain gene rearrangement. We'll come to it. Form the pro B cell. From there, they can form the pre B cell, which undergoes light chain gene rearrangement. Immature B cell is formed. On the right, you see that at this stage, during the pre-B cell stage, you have a mu heavy chain. Mu means IgM heavy chain. So it is the first antibody that is synthesized. And along with that, you have a surrogate light chain. Surrogate means it is something that does not sustain throughout the B cell developmental process, but is initially expressed as a way to keep the antibody stable. From the pre-B cell, we have the immature B cell, where there is a membrane IgM which is present. Membrane IgM means it's an IgM molecule which is present on the B cell surface. All of this occurs within the bone marrow. Now, from the immature B cell, when it enters into the peripheral lymphoid organs, it can encounter its antigen, its cognate antigen, and due to some change in RNA processing, it will form the mature B cell. This mature B cell, why is it mature? Because it now has a predetermined specificity and it has moved out from the site of synthesis, which was the bone marrow, into circulation. So it is ready to face its antigen. But because it has not seen its antigen yet, it is naive. It is not an activated cell. It's not a it's immunocompetent, but it is naive in nature. So at this stage, it expresses MIGM and also expresses IgD, which is from the delta G. Then upon antigenic stimulation, there is interaction between the antigen and antibody, and then that particular B cell gets selected and it forms an activated B cell, otherwise also something you have read as the plasma cell. Then this plasma cell will differentiate into IgM secreting plasma cells immediately, or if required, it will undergo class switching. Class switching means it can transition or change from the IgM subtype into IgG, A, or E. It was also expressing IgD initially, if you see here. So it can then switch into each of these subtypes depending upon what is the pathogen that it is addressing, or rather, what is the antigen that it is specific for. We'll come into details later. You don't need to worry about it right now. But also, apart from these plasma cells, there will be a subsection that will form the memory B cells of all these different isotypes. So this is the entire process that we see here that occurs for B cells. On the right, what you see are different antibody subtypes. So this is the way in which they look, and you can see that the length, the number of constant regions, the specificity, the way in which they're oriented, all of them differ in these different subtypes. The most similar of these, I would say, are the IgG and IgD. But what you see here are differences in the disulfide linkage also. But yeah, most of it is quite similar in nature, I must say. Then comes IgE, which has extra constant regions. IgA, which has a whole other orientation. So it's a dimeric antibody, and it also is associated with something you see here in black, which is a peptide. It is associated with a polypeptide molecule as well. We'll come to the role of IgA, where it is present, why is it needed, all of that. And finally, of course, the pentamer that we had just discussed, and you see how they are oriented also with each other. So in case it is confusing, the easiest way to remember is that this nomenclature has been done based on the Greek letters. So if you know the Greek symbols, they are alpha, beta, gamma, delta, epsilon. So therefore, it has been named as IgA, IgP, IgG, IgD, and IgE. So alpha, beta, gamma, delta, and epsilon. That's how it has been. The nomenclature has been facilitated. Yes. So now we come to a very important concept. If you remember, I had been stressing about it repeatedly in the previous sessions that gene segment rearrangement is something you will understand in great detail, and particularly mentioned the Nobel awardee or the Nobel Prize winner, Susumu Tonegawa, who made this landmark discovery of the way in which immunoglobulins are produced. So as you already have come across in the lecture, that to understand the way in which this insane variety or diversity of antibodies exist, immunologists had proposed two different theories, namely the germline theory, where they said that genome from the germ cells, your eggs and the sperms, they are containing that huge repertoire of immunoglobulin genes, and therefore nothing additional was required to account for antibody diversity. And this was keeping in mind that the theory came out or was proposed at a time when people didn't have knowledge of the human genome and how what is the repertoire of genes that is present in this case. Uh, while this is not entirely incorrect, it's not entirely correct also. And therefore, the focus was also shifted towards the somatic variation theory that the genome has a relatively small number of immunoglobulin genes from which mutation or recombination brings about that huge diversity. So it's actually a mix of two with more stress on the somatic variation theory, which was understood as the mechanism by which the antibody diversity is established. What you see on the right is a schematic representation of what seems like an extremely simple experiment, but what gave rise to the hypothesis that this is how gene segment rearrangement occurs and how immunoglobulin diversity or antibody diversity for that matter is generated. Now you already know that within the genome we have these stretches of DNA which have some specific domains known as B, D, G, and C, variable diversity joining and constant regions of the genome, which are responsible for forming BCRs and TCRs. So what you see, the schematic has basically been described in the legend. So I'll go through both of them simultaneously. This was Tonegawa's experiment. So the experimental basis for diagnosis of rearrangement at an immunoglobulin locus has been shown here. What you see is that when you treat DNA with any restriction enzyme, what is it? If you're not already familiar, restriction enzymes are nucleases. When we say nuclease, it means something which chops DNA, which cuts DNA. Nucleases can either be exonucleases in nature, which means they cut from the ends of DNA, the open ends of DNA, or endonucleases, which can cut within sequence. When we talk about endonucleases, as the name indicates, they are cutting DNA from, like if there's a string of DNA, they can cut anywhere in between. But to maintain the sanctity and the integrity of the genome, these restriction enzymes or restriction endonucleases, otherwise denoted as RE, they have a recognition sequence. They recognize most commonly a 6 nucleotide palindromic sequence, and they cut in between that, or they cut somewhere within that, and they generate some digested ends. So this is called as digestion of DNA, or restriction enzyme digestion of DNA. So if we consider the entire genome and we digest it with some restriction enzyme, you will expect n number of fragments that would be generated, because it would ideally be able to cut anywhere. Like you can see, it would be able to cut anywhere within the genome and would be able to generate small fragments which can be then detected. How can that detection be done? I hope you're familiar with it. There's a technique known as southern blotting. If you're familiar with molecular biology techniques, it would be very helpful. If not, please let me know. I'll have to get into a little more detail about southern blot. But basically, southern blotting is it's a relatively way older technique. Of course, this was done way early on by Susumu Tonegawa. We have much more advanced techniques now. We have different ways of probing DNA. Back then, what he did was basically you have DNA. You take a probe which is radioactively labeled or which is somehow labeled, right? So that probe will bind to one specific region of DNA. In this case, the probe here shown in orange is binding to the J segment of the genome. When it binds, what it will detect is only this fragment because rest of it is getting digested, right? Let me just, yeah. So if you are seeing the restriction enzyme is cutting at each of these sites that are indicated by arrows, you would be able to get DNA sequences here. You would again be able to get DNA sequences here, some here, and so on and so forth, right? You would be able to get short DNA segments in each case. Of this entire population of n number of DNA sequences, the probe, which has been labeled, will only bind to this specific sequence and will be able to detect it. In case of the germline DNA, what he saw was that there was this particular sequence which is being detected at a lower molecular weight or lower basis of DNA. What does this mean? This southern blot is representative of a technique which allows separation of DNA fragment. If a fragment is small in size, it will be able to move farther. Think of it as a sponge, right? Within the sponge, if you add some very explain it, some small beads and some bigger beads, the smaller beads will be able to move farther through the sponge, whereas the bigger beads would be retarded while that movement or while they're carrying out that movement. So the smaller fragment that was present in the germline DNA was able to move forward. However, when there is a rearrangement of DNA, what happens is that the J and V and J segments, or the VDJ segments, or VDJC, etc., all of these segments, they can undergo rearrangement. As a result, when there is rearrangement, those regions are not accessible to restriction enzymes for digestion. So what was the enzyme that was able to cut DNA here previously, upon rearrangement is no longer able to do so. As a result, this fragment will not be generated, and now the probe is binding to a longer DNA segment compared to what it was initially binding to. As a result, this longer DNA segment will not be able to move further, and therefore it will be present much higher, and it will be detected to be as a much larger fragment. So what do we conclude from here? We conclude that some gene rearrangement is occurring, as a result of which restriction enzymes are not able to cut DNA, and therefore we are able to obtain a much larger fragment. That is how he detected that there is some genetic rearrangement or some recombination which is occurring. I tried to explain it in layman terms, but yes, you need a lot of background. You need to know how enzymes work. You need to know how southern blotting is carried out and how detection is carried out and what does it mean. If any of these concepts are advanced for you, please let me know. But mostly, I would presume you would have come across these molecular biology techniques. Uh, so just to be on the safer side, we'll go through the legend once to see what it has to say. So the experimental basis for diagnosis of rearrangement at an immunoglobulin locus. The number and the size of restriction fragments generated by the treatment of DNA with a restriction enzyme is determined by the DNA sequence. The digestion of rearranged DNA, what you see here on the right, with a restriction enzyme will yield a pattern of restriction fragments that differs from those obtained by digestion of unrearranged locus with the same RE. Obviously, we see that for an unrearranged locus, we have smaller fragments because more DNA is available for digestion, whereas for a rearranged DNA, that is not the case. Typically, the fragments were analyzed by the technique of southern blotting. In this example, a probe that includes a J segment is used to identify restriction enzyme digests that include all or portions of the segment. As shown, rearrangement results in the deletion of a segment of germline DNA and the loss of the restriction sites that it obviously includes. Also, it results in joining of gene segments. In this case, it has been represented as a joining between a V and a gene segment that are separated in case of the germline. Consequently, fragments that depend on the presence of this segment for their generation are absent from the rearranged locus. As a result, the rearranged DNA gives rise to novel fragments that are absent from the DNA digest. Again, this might be a little too wordy, but please interrupt me if it is not clear. So this results fragments that are dependent on the presence of this segment for their generation are absent from the digest of the rearranged locus. Simple words, in the rearranged locus, the portion where restriction enzymes could have cut, that region is now lost and therefore restriction enzymes cannot cut it. This can be useful because both B and non-B cells have two immunoglobulin loci. One of them is rearranged, the other is not. Consequently, unless a genetic accident has resulted in the loss of germline locus, the digestion of DNA from a myeloma or normal B cell will produce a pattern of restriction that includes all of those in a germline digest, plus any novel sequence that results from the change in DNA sequence that accompanies rearrangement. So again, this is unnecessary information. It's not required. What I just wanted to bring to your notice is the fact that it's a simple experiment. He cut DNA and he probed it. That's all he did. That was done here. It's schematically being shown for only one region of the genome with only one J segment with only one probe. Of course, this experiment had to be done extensively. You can, if you have some idea of reading research papers, you can go back and you can look at this paper. It's fantastic. You can try to understand what Susumu Tonikawa did and how landmark his discovery was in the field of immunology. But yeah, this was the basic idea of the experiment that was done. So keeping this idea in mind, what we see here is a multi-gene arrangement of immunoglobulin genes. The multi-gene arrangement of immunoglobulin genes has been represented. But before that, what you need to see is the number of segments or the number of the chromosomal locations of immunoglobulin genes in humans and in mouse. So you see that in humans and in mouse, the location of the light chain and heavy chain is on different chromosomes. Um, Somebody might ask why. Why could they just not be present in the same place? Uh, while there can be very different schools of thought, the most logical explanation that I can think of, and what is generally believed in the field as well, is that because this is such a crucial event that it is generating the antibody repertoire or the defense system of a particular individual in case a particular person during development due to whatever unknown reason, if they suffer from some damage in their DNA, if all of the light chain heavy chain genes that were present on the same chromosome, say for example chromosome 14, which has been shown here for heavy chains, if all of them were present there, then what it would lead to is that it would compromise the entire antibody based defense of that particular person. That if there is damage in DNA in one region, it will lead to loss of damage or loss of DNA or some sort of damage to DNA for all of these different gene segments. So it is a way to prevent complete loss of immune defense from an individual. Secondly, as you will look at, as we have seen in the lectures, and as you will see also ahead as we discuss more and more, you will see that this process is very, very heavily dependent upon the process of recombination. So recombination machinery, it looks at sequences, some subsequences, and if all of them were present in the same on the same chromosome, then any V would be able to join to any J or any like the specificity for light chains and heavy chains and for each of these subtypes of light chains that would have been lost or could have been lost. So to bypass all of that, the chromosomal locations have been kept separate so that gene segment rearrangements can occur independently. From there, the products that are generated, they would be able to interact with each other. So that's an evolutionarily smart strategy that nature has put in place. Now, with this background, what you look at on the right is a representation of the gene segments that are present for heavy and light chains. Among heavy chains, we know of the two subtypes, the kappa and the lambda. So you can see the organizational differences also between the two. Again, this is something that has been discussed in the lecture. I don't see the point of reiterating it. Somebody has unmuted.
that it will
So, so I'm not going to repeat the same thing, but just if you go through it, just glance at it, you see that kappa chain DNA has only one constant region, and all of the J regions are clubbed together here. On the other hand, for lambda chain, you see that every V region is interrupted by J regions which are together. And at the front, what you see is the presence of the leader sequence. This is just the DNA sequence which is present. This will all it will undergo the process of transcription and translation, and then it will be directed to its correct location. So while the kappa chains kappa chain and lambda chain are relatively much smaller in size, you see that the intervening DNA is also not, um, it's long, but it's somewhat seems much more reasonable than when we look at the condition for the heavy chain DNA. For heavy chain, you see several different subclasses. Right now, you don't need to get into the details of these. You see that for gamma, you see gamma 1, gamma 2A, 2B, gamma 3. We'll come to all of that. It is the IgG subtypes, not needed right now. But what you see are gene segments for all of the different subtypes of antibodies where the constant regions vary. So you have IgM and IgD, or C mu and C delta, as I have already discussed, to be present first. And that is the probably one of the reasons why the first antibodies during the time of B cell development are IgM and IgD. Following that, you have gamma isotype switching or epsilon or alpha isotype switching, which are present much more downstream. But these are less relevant to us today. Today, we are talking about antibody diversity. That diversity lies within these three. They mediate all of the complicated processes that are the reason why we are able to fight off most of the diseases that we come across. So it is the VDJ or the VJ recombination that we'll be focusing on today. Also, what I want to keep you to keep in mind here are the n number, which is quite different here. So for lambda chain, only two variable regions are observed, V lambda 2 and V lambda 1, obviously. For kappa chain, you see approximately 85 variable regions. So these are all the different V regions that can be seen. And for heavy chain, we see approximately 134 variable regions. So all these different combinations will matter. We'll come to yeah. So we'll come to it in more detail. Now here, it's just again schematic representation of something you're already familiar with, but we'll just go over the process once again so that if there is a doubt, we can address it. Uh, on the left, what you see is a germline kappa chain DNA, which is obviously indicative of light chain gene segment rearrangement. So there, first the joining occurs between V and J regions. Any of the V regions can join with any of the J regions, and therefore VJ joining is giving rise to this first set of rearranged DNA that you can see. Once that has been done, transcription is carried out. The primary transcript contains all of these regions along with the intervening sequences, which we know as introns. From the introns, primary RNA transcript undergoes some post-transcriptional modifications, which includes RNA splicing and polyadenylation. Now, in the process of antibody generation, RNA splicing is probably one of the most crucial processes that you can think of, which gives an antibody its specificity. So because of RNA splicing, what will happen is that this interrupting region would be removed, but also once VJ recombination has been carried out, anything beyond that J and the next C will also be removed so that the second J region does not recombine and does not become a part of the final antibody that will be produced. So after polyadenylation and RNA splicing and translation is carried out, the nascent polypeptide carries this leader peptide as well. And finally, the kappa light chain will be made up of B, J, and C with the variable and constant regions that we are familiar with. I hope it was clear. I went a little fast, but I think the schematics are quite clean and quite easy to understand. But if there is any doubt, please do interrupt. This is an advanced concept. It took me a while to understand it when I had first studied it. But once you understand it, it's fantastic. What you see on the right is a representation of the germline heavy chain DNA. And there, obviously, the first main difference that you see is the presence of a T region or the diversity region. So the diversity region also, once it is present, it will have some role to play. Therefore, its first recombination event is between DJ, that is between any of the D sequences and any of the J sequences. You see the DJ joining, following which we have recombination with one of the variable regions. So there is VDJ joining. Now, this what you see is the rearranged a recombined heavy chain DNA carrying the leader peptide. From this, transcription is completely carried out. You see here the transcription is only carried out till IgD, like till this point, which is why, like I said, the first antibodies to be produced carry the IgM and IgD isotypes only. If required, they will undergo isotype switching, but that occurs much later. From this transcribed DNA, you have from the DNA that underwent transcription, you have the primary RNA transcript, which undergoes the same process, polyadenylation or splicing. And upon splicing, it will either undergo this first variant of splicing, where IgM will be kept, or it will undergo the second variant of splicing, where IgD would be kept. In either case, you will have it will undergo translation, and you will have either the mu heavy chain or the IgM heavy chain, or the delta heavy chain or the IgD heavy chain. Based on what is produced, the antibody subtypes will vary. Now, these heavy chains will form an association or will form a dimer with light chains, and then the dimer of dimers, which means that heavy chain light chain and heavy chain light chain will associate with each other and finally form the antibody, which is what we are familiar with. This reminds me that you might this is also often proposed as a question that which of these following molecules is a dimer of dimers. So antibody is a dimer of dimers. The first dimer is between a heavy and a light chain. The two heavy light chain combinations, they associate with each other and form a dimer yet again, which is known as a dimer of dimers. So this I showed you just how the multi-gene arrangement of the antibody loci is how it is present within the system. Now, is that all? Just having the diversity and just some processes of some combinations and some recombination, is that enough or not? Excuse me. So we saw that this representation of the different gene segments that are present, that actually proves the germline theory as correct. But we had extensively discussed that that's not enough. There are multiple sources of antibody diversification, which has identified not just being in humans, but also in mice. First source of diversity is the presence of these germline gene segments. Second, like I said, is the combinatorial VDJ joining. Apart from that are several processes which we'll discuss, but also the combinatorial association of heavy and light chains, which gives you an extensive amount of diversity. If you remember the lectures by Professor Shruti, you would have seen that he multiplied all of these different combinations and showed that it did give rise to a few lakh combinations, but we have a lot more. We have approximately 10 to the power 5 range of diversity. So for that, several other processes also come in. What does that include? You have junctional flexibility. This I don't think has been discussed in the lecture. I will briefly be discussing it today. P addition and N addition, which is something that has been discussed, that is P region nucleotide addition and N region nucleotide addition, somatic hypermutation, and of course the combinatorial association. So these are the 7 sources or the 7 means for antibody diversity in humans and in mice. Again, I am speaking a lot. If there is any doubt, please interrupt and ask me. And if it is too fast, please let me know that. Also, I'll try to mellow it down somewhat. So yeah. Now, what we look at here is the way in which this P and N nucleotide addition is actually brought about. So what you see here is a representation of some sequences or some consensus or some signal sequences. Let's just call it that. Some signal sequences that are present in this immunoglobulin DNA. If you look at the top, this shows the nucleotide sequence of RSS or recognition signal sequence, which already has been discussed by Professor Shrudeep in the, I think, third lecture, if I'm not wrong, third or fourth lecture for this week. So there you have 2 stretches, a nonamer and a heptamer, which actually gives rise to the signal sequence because it is interrupted by a stretch which contains either a 23 base pair fragment or a 12 base pair fragment. When there is a 12 base pair fragment, then there is a nonomer, which is a stretch of 9 residues, 12 bases, and again a stretch of 7 residues. This is called as a one-turn RSS, or one-turn recognition signal sequence. It's just an easier way of representing that DNA segment, which is known as the one-turn RSS. On the other hand, when there are 23 bases that are between a heptamer and a nonomer, as is shown on the right, that is considered as a two-turn RSS. So these are just to simply represent it. They have been demonstrated as little arrows, but they effectively represent this entire stretch of DNA sequence, and this is relevant because these signal sequences can only communicate with each other in a certain manner. How is something we'll look at now. Let us go over the legend. The first part A, shows signal sequences which consist of some conserved heptamers and conserved nonomers, which are separated by non-conserved spacers. When we say non-conserved, it means that they do not have any consensus sequence. Any nucleotide can be present there. And that non-conserved spacer can either be of 12 base pairs or 23 base pairs. And on the basis of this, the 1223 rule had actually been proposed, which has been discussed by Professor Shrudeep in the lecture as well. In figure B, you see the two types of RSS designated as one turn and two turn have some specific locations within the chains, within either the lambda chain or the kappa chain or the heavy chain germline DNA. During the DNA rearrangement, gene segments that are adjacent to the one-turn RSS can only join with segments adjacent to the two-turn RSS. So what you see here is for the lambda chain, you see that after the variable segment, we have a two-turn RSS. And for the J segment, we have a one-turn RSS, and only these two can join with each other. Why is this relevant? It is because if we look at the heavy chain DNA, that's where it becomes most relevant. That if variable segment has a two-turn RSS and the joining segment has a two-turn RSS, then they would not be able to recombine with each other, and instead they would combine with something which carries a one-turn RSS, which is the DH region. So first the D and J region would undergo recombination, then the V and DJ fragment will undergo recombination. So that's how this is kept in check. And how does it operate further? So this is a model that depicts the general process of recombination and immunoglobulin gene segments, which has been shown with V and J only, just to keep it simple. So you have already looked at it. There is deletional or inversional joining. I forgot to highlight it. But yeah, the two ways in which the process of immunoglobulin generation can occur is either through deletional joining or through inversional joining. So in case of the deletional joining, you see that there is a recognition signal sequence which is present, which is identified by RAG1 or RAG2. RAG1 is the is a recombinase which facilitates this process. There is synapsis. Synapsis means the alignment of homologous regions. So there is a search for regions of homology, and once that is found, alignment has occurred. Then there is single strand cleavage by RAG, and you can see that upon cleavage, these regions are opening up. Once it has been cleaved, then there is formation of a hairpin and a double strand DNA break that occurs. So these are the hairpins that are formed, and that is the result of a double strand DNA break, which is mediated and controlled by RAG1 or RAG2. Once this has been done, what happens next is that this entire region will get excised as the signal join. The rest of it will form the V. The rest of it will what you get out of it is that these two ends, this one turn and two turn, will combine with each other and will form a covalently closed circle that will be eliminated out. That is called as the signal joint. On the other hand, what remains behind is that this V region is joining with this J region and it is forming the coding joint. Coding joint simply means something that will go ahead and will be coded into a functional RNA and then protein. So that is what is formed there. On the right, the process of inversional joining has been shown. And inversional joining means that something is being flipped around, right? When that is occurring, you see that these are facing the same direction. Both the one-turn and two-turn RSS are they're facing opposite directions. They are looking in these opposite ways, and whereas for same direction, these blue arrows indicated same direction, they would be able to coordinate and they would be able to excise the intervening fragment. In case of inversional joining, we see that the process occurs in a similar manner, but instead of getting excised, what happens is that there is the uh, cleavage of the hairpin by endonucleases, which will generate sites for p nucleotide and n nucleotide addition. I'll come to it later. But instead of getting excised out, what happens is that there is repair and ligation to form a signal joint and a coding joint, which is being retained. We will just go through the legend so it becomes a little more clear. So deletional joining will occur when the gene segments to be joined have the same transcriptional orientation indicated in blue arrows. This process leads to products, a rearranged VJ unit that includes a coding joint, which is removed, and a circular excision product, which consists of recognition signal sequences or RSS, signal joint, and intervening DNA. In B, the inversional joining is observed, which occurs when the gene segments have opposite transcriptional orientation. In this case, the RSS, the signal joint, and the intervening DNA, all of them are retained. And the orientation of one of the joint segments becomes inverted. Both cases include some addition or deletion that occurs before the sequences are finally rejoined. So this is the way in which antibody diversity is being regulated at a molecular level, that either there can be excision or there can be inversion of subregions. Yeah. So that was the way in which this recombination was actually facilitated, and the way in which this is carried out, this entire process of deletional inversional joining to finally allow recombination to proceed, is a major contributor towards the generation of antibody diversity. Apart from that, one particular topic that was not really looked at is the concept of junctional flexibility. It's not a very complicated topic as well, at least not at this stage. You can just fundamentally understand what is happening there. So junctional flexibility, as the name very directly, oh, sorry, very directly includes, it is about some uh, less stringent way of junction formation, or some flexibility in junction formation. So what you see here is a representation of junctional flexibility in joining of immunoglobulin gene segments, illustrated with PK and JK. I'm reading out the legend from here. In phase joining allows a productive rearrangement, which is translated into proteins, whereas out of phase joining is what it leads to non-productive rearrangements that contain stop codons and is effectively not translated into protein. So when does this occur? This occurs when you have this B region for kappa chain, and here you have the J region for kappa chain. When recombination actually is occurring, it is um, nobody is able to exactly see where that recombination is occurring, right? None of the enzymes are able to do that. They are just looking at DNA, finding some weak interaction between them, some regions of homology between them, and it will join. So when we look at all of these different options, 1, 2, 3, 4, and 5, you see that in each case, the junction is breaking off at one point and joining at another. Let's look at this first representation of junction formation. It is breaking off, B region is breaking off between C and D, and it is then joining here, right after GTG from the JK. So after three amino acids, you can see insertion which is occurring. So as a result of this rearrangement or this VJ joining, what will then be produced is that glutamate and aspartate will then bind with or will then connect with some base which is present as part of the J subunit, will then lead to formation of threonine and arginine amino acid residues. So this is an in-frame insertion or in-frame recombination which is occurring, which is why it is leading to or is considered as a productive rearrangement. On the other hand, if it leads to the generation of some stop codons, etc., that there is some that somehow how do I put it? That while the rearrangement was initially supposed to be helpful and was supposed to bring more helpful antibodies into circulation, instead what it is doing is the sudden appearance of a stop codon is leading to the production of a truncated or a partial protein. Now, this partial protein will be incorrect, will not form the right sort of antibody, and will be, responsible for more harm than good. So that is the reason not every rearrangement is always correct. There is flexibility, and only those rearrangements which are productive will be retained further. So yeah, this is another source of diversity, because depending on how the junctional association is being carried out, very different combinations of antibodies can be observed, and as a result, that is leading to more and more diversity. So this table actually shows a comparison of the different types of uh, germline segments and the way, or DNA segments that are associating with each other in a way which is bringing diversity. So this table actually also had information about mouse. I did not think it was relevant, so I did not include it. If you are interested, please go back to the book. These are all part of Kubi, so you can refer to Kubi for any conceptual difficulty that you might face at any point of time, because it will have information 90% of the time. So yeah, what you see here is a representation of the different frequencies of germline segments B, D, and J. Estimated number in humans and in the heavy chain, and estimated number in the light chains have been shown for each of the gene segments V, D, and J. The combinatorial VDJ and VJ joining will show you the possible number of combinations. For each of the subtypes, you can see that there is a difference in the numbers that are generated. So that is showing you approximately 8262 combinations for just the heavy chain by itself, and approximately 200 and 120 for the light chain by itself. Taking all into consideration, you are seeing a number of possible associations. But that also is not enough. There is even more diversity that is brought about, which is something we'll come to in the next few slides. So what you see here in the table at the bottom is um, the source of variation that is coming about uh, within within different regions of the antibody epitope itself. So yeah, somebody has a question.
Slides has not changed. It is
I
showing
have
that
not. Yeah, what can you see? Tell me.
it is showing the generation of antibody diversity.
Yeah,
I mean,
I'm
you are
explaining
talking.
that slide only. And here, source of variation that you see: CDR1, CDR2, CDR3. I'm explaining that part.
No,
We'll
ma'am.
move on.
No, ma'am. It is showing this 7 times of generation of antibody.
Oh, is
Only.
it? Is everybody facing that difficulty? I've been explaining it for quite a while. Uh, can anyone else please unmute and let me know if you've been able to see slides all this time?
Yes, ma'am.
Okay, maybe Soumya, you were facing some difficulty. I'll just present it once again. Just a minute. I hope it is visible now. Maybe. Soumya, please check from your end. I think it could be some internet issue. Don't worry, you will get this video uploaded. So you can just go to this part, like around 1 hour, 15, 20 minutes later. You can read up on that part
Okay,
if
ma'am.
that's difficult. Yeah. So this is, I hope you can see it now. So this is what I was talking about, the source of variation. So we talked about the epitope of the antibody, and within the epitope, there are hypervariable regions, or the CDR1, CDR2, and CDR3. Among these three, the CDR3 is considered to be the most relevant, or the most responsible towards contributing for the antibody diversity. So there, the sequence gets encoded by not just V segments, but by VLJL junctions and by VHDH junctions, which is why it has maximum variation as part of the sequence. So this variation is also accompanied by junctional flexibility, which is present, P and N nucleotide addition. N nucleotide addition only occurs in the heavy chain DNA. It has been given as a disclaimer, and also somatic hypermutation. So while within CDR1 and CDR2, only somatic hypermutation is bringing diversity or variation, CDR3 has all of these, and therefore it is most relevant towards contributing for antibody diversity. Now, what are these P and N nucleotide additions? Again, this has been discussed by the professor in the lectures, but we'll just go over it very briefly. Again, these are just more schematics for you to understand the same concept better. So on the left, you see P nucleotide addition, and on the right, a nucleotide addition can be observed. So what you see, if you remember this process, I don't know if I have the schematic for it or not. But yeah, basically deletional or inversional joining was occurring, right? So that was facilitated by the recognition of the signal sequences by RAG1 and RAG2. And once these cuts had been made, then here you see the formation of hairpins. That after double-stranded DNA cuts have been made, or not after, it's actually a simultaneous process, that once DNA cut have been made, the two ends of that um, now linear DNA are ligated with each other. And this is where the site, this is where what is considered as the site for P and N nucleotide addition as well. So that is the part we are focusing on. And here you see, okay, this is actually the last slide for today. Okay, so that's what we are seeing here. So you see that there is this D region and this J region, after which just a few nucleotides are present, and that these two have been joined to each other. Why is it necessary to join these? This is because it prevents it from being immediately digested, and it keeps the DNA molecule stable for that duration, so that any exonuclease doesn't just come and chew it up and completely remove the DNA that is encoding antibodies within that cell. So these two ends are ligated with each other, and then surprisingly, without cutting into the hairpin, the ends of these regions are actually cleaved upon for generating sites for T-nucleotide addition. So here you see some cleavage which is occurring, cuts that are generated. As a result of those cuts, you see it is now linearized, and instead of 2 nucleotides, you see the opening up of 4 nucleotides. So basically, once here it has been cut up, that DNA sequence, which was initially ligated, is now just opening up. This is what is happening. So as a result, apart from these first 2 nucleotides, 2 more have now been added. What then happens is that more and more repair enzymes will come in, and they will start adding nucleotides randomly. After addition, what happens next is that after addition, there can be, okay, we'll come to that later. Let's go through the legend first. If the cleavage of the hairpin intermediate yields a double-stranded end on the coding sequence, then P-nucleotide does not occur. P-nucleotide addition does not occur. However, in many cases, cleavage often yields a single-stranded end. During subsequent repair, complementary nucleotides are added, which are called as P-nucleotides, to produce palindromic sequences. That is what gives rise to the P within this nucleotide addition. This is indicated by brackets. So what is happening here is that if you have a double-stranded end, that if the cleavage is getting, if the hairpin is getting cleaved in such a way that you have a double-stranded end, say suppose it was getting cleaved here and you got a double-stranded end, then P-nucleotide addition will not take place. If it does not happen in this way, and instead you have this stretch of sequences which opens up, some nucleotides can be added which will be complementary to each other. As a result, the effective sequence will become its own palindrome. It is TCGA this way, it is TCGA this way. It is TATA this way, it is TATA this way. These are two separate palindromes. In these examples, 4 extra base pairs shown here in blue have been added to the coding joint as a result of the P-nucleotide addition. So that is another source of antibody diversity, that while the initial sequence did not have these nucleotides, these are getting added now. On the other hand, besides P-nucleotide addition, some random nucleotides can be added. Keep in mind, this random nucleotide addition, otherwise known as N-nucleotide addition, will only occur for the constant chain. this in mind. Here you see that constant chain, it happens in the heavy chain. So N-nucleotide addition can only occur in the heavy chain and is responsible for diversity of the CDR3. But P-nucleotide addition, it can occur in the light chains as well, but does not occur for CDR1 or CDR2 in either case. It's a lot of information I know, but that's why I try to put everything in tables so that you can, organize all the information in your head for your own clearer understanding. So when there is addition of some random nucleotides apart from these, so till here, if the process is stopped, then it is just p-nucleotide addition. When more and more random nucleotides are getting added, which are not helping build a palindromic sequence. Say, for example, here 3 more nucleotides have been added, but as a result, what is generated is not palindrome in nature. Then in that case, these are called as N-nucleotide addition, and this has been added by the terminal deoxynucleotide transferase or TDT, and it does so when there is joining of heavy chain coding sequences. So what you are understanding from here, if you go back to this table once again, for the heavy chain, initially we had more number of gene segments for both V and for J, and additionally, there were some diversity segments that were present. Now what we are seeing, there are 2 more processes that are adding on to the diversity of the heavy chain, which is coming about as a result of p-nucleotide addition and n-nucleotide addition. Light chains, apart from this VDG segments, they also are undergoing p-nucleotide addition, but not n. But along with all of this, each of these segments during the time of recombination can undergo this entire process of junctional flexibility, which is also adding to the diversity. So taking all of this into consideration, let me just go back to that process. Yeah, taking into consideration all of these steps, what we can come as a conclusion to is that there are all these means of antibody diversification. First, presence of multiple germline gene segments. Second, combinatorial joining of V, D may or may not be present, and J segments. Third, junctional flexibility, that when a V and D region are joining with each other or they're undergoing recombination, that exact nucleotide sequence where the 2 junctions are binding with each other can actually be different in different cases. Only when productive rearrangements are formed as a result of which a functional protein will be generated, only those are tolerated, but they also bring rise to more and more diversity within the antibody repertoire that can be taken into consideration. Apart from that, we have P region and N region nucleotide addition that can occur. And there is somatic hypermutation as well. Somatic hypermutation, I don't have in my slides today, but basically it is a process where even more variability or random mutations can occur within the somatic cells, within the antibody epitopic regions, which are responsible for binding to the paratope of the antigen. Random, non-specific mutations can occur. Hypermutations means they are occurring at a pace which is much, much, much higher than normal, and they are occurring in somatic cells. And finally, the combinatorial association of all of these different combinations. We have the raw material for antibody generation that by itself, just by itself, is so huge in diversity that when you take into consideration the combinations of all of these, the entire antibody repertoire that is generated is immense. So all of these together have been studied as the means of antibody diversification. Please keep in mind the exact numbers might be a little outdated. The book is quite old. It's from the 5th edition. So I still don't have access to the current edition, so I was not able to get it. If there are any changes, I'll go back and check it once more. But this is the overall idea that has stayed constant on all of this time. So that was the material I wanted to discuss for today. If there is any doubt, please ask me and we'll go over these questions and then we can end the class for today. So I'll just go over the questions in the meantime. If there are any doubts, please hold on to them and then bring it up and we'll try to address it. So what you see here are some just sample questions from previous years. So the first question talks about the antibody effector functions. They include all of these following, except first of all, they activate complement on bacterial surfaces to promote phagocytosis. This is true. Complement activation is something we will get into eventually. Second is binding of extracellular viruses to block their entry into host cells. Third is binding intracellular viruses to initiate cytotoxicity. or blocking uptake of bacterial toxins by host cells. So antibody effectors can do all of these. They can activate the complement system. They can bind to extracellular, something which is outside of the cell. The antibody can bind. It cannot enter cell directly. So intracellular viruses, it cannot bind and it cannot initiate cytotoxicity by itself. Therefore, option C is the correct answer. Uh, Second question is about which of the following is responsible for the diversity of the hypervariable region of the immunoglobulin? A, the hinge region, CDR, epitope, or aggregope. Aggregope is something which you can come to a little later. You'll have to understand the way in which aggregation, agglutination, all of these processes occur. We'll come to it later. But just for now, it's a terminal. It's an interesting terminology. So maybe for now you can Google it, but conceptually we'll come to it a little later. For now, we are talking about the diversity of the hypervariable region. So that is facilitated by CDRs or complementarity determining region. The hinge region is mainly responsible for holding the two fragments together and it's necessary for the solubility of the antibody. And of course, the epitope is responsible for binding to antigenic parental. The third question is about the exon encoding the V region of an immunoglobulin protein is generated by a process of somatic recombination. This recombination event brings V and J segments together, where A, in all cells of the body to encode a V region sequence, no, to generate maximum diversity in the CDR3 sequence of the V region. If you remember that table once again, CDR1, 2, and 3 are being compared. So this VJ and VDJ junction, it just in case it's not clear, this table is what I'm talking about, that this recombination only occurs for and it's only hugely contributing to the diversity within the CDR3 or complementarity determining region 3 or hypervariable region 3 of the V region. That's the correct option. The remaining options were about the alternative splicing to encode a V region sequence. No. By a precise mechanism that never adds or loses nucleotides at the junction. Also not true. We have talked about P and N nucleotide addition just now. To generate a single exon encoding the entire globular protein. That's also not true. So the correct option is option B. The next question is about the digestion with pepsin. So if you remember the first slide, I had shown this representation of how pepsin cleaves, and this is where pepsin is generating a cut in the antibody. So pepsin digestion of immunoglobulin G or IgG, the first antibody representation is of IgG only. So what does it generate? FAB, two fragments, and low molecular weight fragments, two Fab fragments, and FC fragment, one heavy chain and one light chain, or only light chains. So if you remember this cut site for pepsin, you would know that obviously only light chains cannot be generated because the disulfide linkages are intact. One heavy and one light chain is also not correct because then it would have to cut at the hinge region. Two Fab fragments and FC fragments is also not correct because then it would have to cut above the hinge. But because this cuts, pepsin cuts below the hinge, what it will generate is the fragment with two antigen binding sites and the lower constant region, which will be a low molecular weight fragment. As a task from your end, I want you to go and check in which case the rest of the three options would be correct. What enzymes or what chemical treatment is able to generate each of these different. These will all be these are. It's possible to take antibodies and generate all these different types of fragments, only Fab fragments or only heavy chain, only light chain, one heavy, one light chain. It's possible to get all of these. So I want you to go and check up on it. If not, we'll discuss it in the next class. It will be interesting. You'll be able to understand how experiments are done to understand the structure of antibodies. This is important because crystallization as a technique or cryo-electron microscopy or all of these techniques have come up much later. They are obviously the short way of knowing the structure of any system. But simple molecular biology can delineate the structure of immunoglobulins like that. It's just a few experiments. So having data and understanding data are two completely different things. So if you will look into the way in which these fragments can be obtained, it will help you really understand how it will enrich your scientific temperament is all I can say. The next question is about the class of immunoglobulins is determined by A, pH region of the heavy chain, CH region of the heavy chain, light chain, or the carbohydrate attached to the light chain. So we talked about the different subclasses or the isotypes of antibodies or immunoglobulins. So that is obviously determined by the constant region of the heavy chain, where you can have IgG or IgM or IgD initially, and then you can undergo switch into either G, A, or E, depending on uh, what is the specificity for. So option B is correct. And finally, immunoglobulin IgE differs from IgA in A, the number and location of interchained disulfide bonds, the number of C domains, the length of the hinge region, or all of the above. So if you remember, I had shown a comparative structure of all of the different antibodies. So it actually depends. Let me just go back here. Yeah, so this is IgE, which you see, and you can see its constant region is different. Its disulfide linkage is absent between the two dimers. Also, the length is different, so it differs in all of these things. So option 4 is actually correct. So number and location of interchain disulfide bonds is different. The number of constant regions is different. The length of the hinge region is different. So based on that, fourth option D is correct. That IgA differs from IgA. All of these different aspects. So with this, I have reached the end of the material I wanted to discuss today. Um, Now, in case anybody has any questions, please go ahead and ask, and then we can end the class for today. I hope you understood. There were a lot of concepts. Most of it has obviously already been discussed, but I wanted to get a little bit into the experiments that can help you understand how these were done. So if there are any questions, please raise your hand. I'll try to help you out. Okay, if not, you can maybe upload it somewhere on the discussion, or you can mail me. And if the slides are not accessible by next class also, then please let me know, because these are uploaded as soon as the classes are done, either today or maximum by tomorrow, I'll upload it. If it is not accessible, please let me know in the next class. And yeah, go through the slide materials, not just mine, but if possible for the other days as well. It should be helpful. Um, and yeah, please come up with doubts. We want to have interactive sessions. Otherwise, it just seems like another lecture to attend. It'll get boring for you as well over time. So please come up with questions, doubts. If you have some idea that you as a scientist can think of, we can discuss those as well. And yeah, thank you so much for joining. I hope it was educational and you could understand what I was trying to convey. But yeah, I hope you have a nice day, and you will have access to all of the materials soon. So thank you so much for joining. I'll end the meeting for today.